So tonight we have Stephen Jones, who is our arborist, and Dr. Susan Pell, who is our Deputy Executive Director, telling us all about the corpse flower in peak bloom. So take it away, Stephen, Susan.
Hey everybody, we're so happy to have you here tonight virtually to see this wonderful, amazing plant in peak bloom here. We've got the spade really opening up, and it's extremely stinky, so this is one of those rare occasions when I'm super excited to have a mask to be able to wear it because it's pretty stinky. We're going to get up on the ladder here in a minute, and that's a treacherous situation to be in. But I'm going to turn it over to Steve, and he can tell you a little bit about this Amorphophallus titanum.
All right, so we have a plant here that we were donated from seed. We've grown it for 8 years now, and it's blooming for the first time. So for the first time, we get to see its individual characteristics. These plants are very highly diverse individually because of the cross-pollination requirements. So this one turns out to have a very dark purpley inner spathe, and the spadix, this structure at the top here. It has kind of it's a greenish yellow color, and there's a little bit of purple coloration as well in there. So another unique individual for us.
Yeah, and I would say this is probably my fifth one of ours to smell, and it's very sulfury, quite a bitter kind of a smell. Like a really, really big dead mouse. quite nasty actually. So Steve, you want to talk a little bit more about when did we start noticing this opening up today?
Well, I noticed it about 3 o'clock, which was too late for me. I was a little disappointed. I missed it by about 20 minutes, I think. But earlier this morning, we did see a piece of the spathe start to fold back away from the spadix. And then about 2:33, it really started to open up. And we were able to see that pretty obviously on the webcam. So once it got started, though, it really got going. And it's still going. It's still on its way open.
Yeah, so it's not peak yet. It still has some more to go.
A little bit. I mean, I've been measuring it about every half hour, and right up to this past hour, it's been still on its way open. So, not sure how far it'll go, but some of them open much further than this. And we'll know when it's fully open that the female flowers are presented at that point.
Yeah, each one is really individual, so it may open up quite a bit more and have more of a flat appearance at the top of that spade, which is that large maroon and green leaf-like structure underneath the big tall spike in the middle, which is called a spadix. And so it may open up quite a bit more. It may stay a little bit like this. Just to kind of, for anybody who wasn't with us earlier today, I did a program this morning at about 11, and at that point, it wasn't heating up. The spadix wasn't heating up yet. It didn't smell yet, but the flies were already being attracted to it. So I couldn't smell it with my human nose, but the flies were already sensing some chemical changes happening in that inflorescence.
Yeah,
Yep. Go ahead.
There's a number of flies still on the outside of the spade, and I've seen them flying in a little bit on the inside, but yeah, that's kind of been picking up the activity there, and I'm sure we'll see even more later.
So we got a couple questions here. What is the height, and is the spadix on the plant purple?
We were at from the top of the corm, so which is a little bit of a guesswork. We dig in there and try and find it. To the very tip of the spadix, we're about 102 inches this morning, and that's really it. Once it started opening, it's done with growing upward from what we could tell.
This is our tallest bloom to date that we've had.
Are there other bugs except flies that are interested in it?
Well, not in the greenhouse, not in the conservatory. But yes, anything out in nature in the tropical forest, you'd see all sorts of insects. Carrion beetles are a big pollinator of this plant. But basically anything attracted to a rotting animal out in the jungle would come and visit this inflorescence.
And can you really describe what the smell smells like to you personally right now?
What do you think, Susan? For me, it reminds me, I used to work on the golf course, and it reminds me of a pesticide I used to spray on it, and I hated it. And it reminds me of that. It's awful. It's the bottom line.
Yeah, to me, so I actually, another garden that I used to work at had one of these bloom, and we had the scent analyzed at a professional scent company, a Japanese company, and there's mostly sulfur compounds in this scent, and this one has a lot of really heavy sulfur compounds. So if you've ever like struck a match and you smell that smell right after you struck a match, that's sulfur. That's a really light dose. If you imagine striking, I don't know, several hundred matches maybe all at once, that's the bite that you get in your nostril when you smell this thing. So it's sort of between, a dead animal, a trash truck, and just straight up sulfur.
And if you've ever been to a sulfur spring, I'm from upstate New York, and there's sulfur springs there. It's similar to that. I'd say this is a little worse, a little more unpleasant.
A little trashier.
Yeah.
Awesome. Thank you for that description. Someone was asking about the difference kind of between the two, noticing that the other plant, the second corpse flower, has a purple interior versus the green one on the right.
Yeah, and there's a lot of diversity within this species, that you get a lot of different color changes, color diversity within the spadix, that large pointy thing that comes out of the middle there, and also the spade, the big leaf that subtends it. So those go anywhere from a really, really dark purple into sort of maroons, over all the way to a really light yellowish green on both the spade and the spadix there. Just like you think of, tulips have lots of different colors in their petals. Same thing here, just lots of different colors.
So what are the plans now? Once it reaches peak bloom, once it fully opens and it's like at its stinkiest, what are the plans what plans do the Botanic Garden have for that plant? We're pollinating it. What are we doing with it?
Yeah, we were lucky enough to have some pollen sent to us from Chicago Botanic Garden. They're helping us on a conservation project. They're leading a conservation project with this species. So they have pollen stored from 3 individuals actually that are unique to these. And so our plan is to cut a window into that space where the flowers are located. That's towards the base. And we'll cut a window in there, and we'll take a paintbrush and brush the pollen onto the female flowers. And we'll try and get about 40 to 60 seedlings that will be a whole group of new genetically individual plants. And there you could see Curator Bill 3 years ago pollinating actually the parent of this second inflorescence that we'll have blooming here in probably a week and a half.
When would that be happening, the pollinating?
Our plans for early in the morning, about 6 in the morning. The flowers will be receptive somewhere in the middle of the night. But that period where we were able to pollinate them goes much longer than that, for a number of hours. So we'll wait till early in the morning and get up and get going.
So yeah, anytime between about 12 and 24 hours, you have receptivity in the female flowers, which are the first flowers to mature within this inflorescence. So they'll be ready for pollen, and then they will stop being receptive. So you can see them there. That little sort of yellow tip there, that's the stigma. That's where the pollen is going to land. And that connects down through a style to that pink ovary at the bottom. The ovary is going to turn into the fruit if these are successfully pollinated. And those are going to be receptive first, as I said, and they're going to no longer be receptive to pollen. They're basically finished with pollination cycle, and then the male flowers will go into their productive stage, and they'll release pollen. So after the females, and we'll collect that pollen and we'll share that with other botanic gardens, or possibly cryopreserve some of it.
Right.
Yeah, if you pull up a picture of the male flowers, you see those. Male flowers are located above the female flowers in the inflorescence. So there's basically, inside of this long spadix here at the bottom of that spade, you can see that part of the spadix that we can see emerging, it sort of kind of tucks in, and it's just right above those male flowers. You can see it in that picture there. And then below the male flowers are the female flowers. And so sometimes people will see these things after the male flowers release their pollen, and the female flowers will be covered with pollen from that same inflorescence. But remember, those female flowers are no longer receptive by the time the pollen is released within the same inflorescence. And what that does is it prevents self-pollination from occurring within these plants. So that increases the likelihood of genetic diversity, increasing genetic diversity by cross-pollinating.
And so there was a question, what pollen are you guys using to pollinate from? And are you tracking that? I'm sorry if you were already talking about that a little bit, but
yeah, we are. So for this one, for our female flowers, for this one, we've got pollen from Chicago Botanic Garden. And then we are also working with Huntington to exchange some pollen potentially with them. They've got a bloom that's going to be right on the heels of ours probably. And so if ours goes first, then we'll collect our pollen and send it out to them. And then vice versa, we'll probably get some pollen from them to pollinate our second bloom.
Awesome. Anything else that's interesting about the plants right now in their current state?
Really, just the stench stands out at the moment, to be honest.
And that does it pulse? Doesn't that something that happens sometimes where the stench like it's worse and then backs off a little bit?
Yeah, so we've got some anecdotal evidence of that in the past, and I have measured tonight. I've been measuring about every half hour the temperatures along that spadix, and they have varied by half a degree to 1 degree, and so there might be a little faulting there. I'm not sure if that coincides with the stench, but yeah, it's something that might be happening.
Yeah, and that's pretty standard in blooms that heat up. So this ability to produce heat is not unique to the aroid family. Certainly many aroids do it, but we also see it in a lot of other families as well. So the magnolia family, for example, their individual flowers heat up. You get, there's a lot of different ones. There's a lot of gingers that heat up, and they're for different purposes. So some will heat up to melt ice or snow around them. Of course, not what this one's doing. This one is heating up to volatilize those sulfur compounds. And so the reason that we are standing here, 6 feet away from it, and it smells really bad is because it is heating up. I gave this example earlier, but this is like when you're chopping garlic in your home, in your kitchen. If you're just chopping that garlic, nobody else can smell it in the house, elsewhere, unless they're really close to you. But as soon as you put it in a frying pan, the whole house can smell it because you volatilize those compounds in the garlic. Same thing here, it's heating up, and it's going to spread those scents in the wild about a mile away if we track pollinators from that far away.
Awesome. Someone asked, in the wild, do they all bloom around the same time? It must be hard for them to pollinate each other, although bugs would help.
Yeah, so they don't all bloom at the same time. They're going to be spaced out a little bit. Just like if you think of, anything that's any of our native plants that we're blooming here, they're going to bloom in the same relative time period. So, over the same month or so, but not, all on the same day kind of thing. And so the likelihood that there's another, originally before the distribution of this plant was sort of disturbed by development and whatnot, then there's a pretty good likelihood that another individual of the same species would be going into bloom in a complementary cycle. So they would have pollen. The pollinators would already have pollen on them from another bloom when they came to be attracted to the female flowers from, when it was emitting its odor. And then they would get the pollen from that bloom and go on to the next one.
You mentioned this a little bit in that answer, but are they endangered at all?
Yeah, they were recently listed as endangered plants. Actually, what's this? The status is near threatened.
Endangered. I believe it's endangered.
Endangered. Okay.
Yeah.
Yep. Yeah, that's their conservation status is endangered, and that's due to mostly to habitat destruction.
Someone in the chat would like to know how far apart the two plants are behind you. They look like they're fairly close together, but distance-wise, how far apart are they?
Close to 6 feet, 5, 6 feet.
Yeah, at the base. So that spade, because the spade has opened up so much, then it's only like 2.5 feet, probably something like that, 3 feet away now. But if you look at the center, from center, it's about 6.
So they're following good social distancing protocols.
They are, yeah.
Good.
That's right.
That's awesome. Yeah. I'm gonna hold on.
Taking social distance from them by all people as well.
How wide is that spade?
The spade, that's about big.
The last I measured an hour ago was over 3 feet, so it's probably 3.5, I would guess, maybe more.
Yeah, 4 feet.
And do you anticipate it getting much wider than that?
Again, it's kind of it's guesswork. Nobody's seen this individual bloom before, and they do tend to have individual qualities as far as things like that go. So that's one of the reasons I'm measuring it. I'll know when it stops, but at this point, it seems to be continuing to open. And like Susan mentioned before, some of them will go completely, almost like a dish, completely perpendicular, whereas some will stay more tulip shaped.
Yeah, and one thing you might not be able to see in the image is that this spade is actually not not like a continuous fused leaf. It's actually open all the way down to the bottom on one side, and so it really has this ability to not just sort of open up by spreading there, but actually can sort of split in the front and open up even further. So it's not fully fused. It's not a tube. It's not a solid tube. Yeah, you can see there a nice image where you can see that opening part where there's overlapping tissue on the spade. Yeah, and I will say this one, what's kind of nice about this pair is that they're really nicely matched color-wise. So the spade and this one, this is this really beautiful dark maroon, and it's really kind of matching the spadix on the other side. And its spade right now anyway is still that nice kind of cabbage color, nice and light green, like the spadix is here. If you can see really up close on this, the spadix is actually quite interesting. It's sort of a kind of a green with almost like a little purple on top of it, and then it has tons and tons of little green dots all over it. And you can see that in that nice picture there. It really has a purple hue to it with that underlying green color. Really beautiful. One of the things that we sometimes do when we're trying to breed these, we'll select pollen from a plant that has a really different flower than the one that we're pollinating to try to get some diversity in the colors there. This is a very common strategy in cultivating plants in general to breed for diversity and to see what you get in the mix.
So I know we have kept track of height. Like this is the tallest one we've ever had. Have we also tracked the width at all in terms of, or is that something we're looking to track at some point?
We've got the width tracked now. Like I said, I think this one's closer to 3.5 feet right now, maybe a little more than that. I'm not sure of historically what we've measured there. There is a lot of variation though, so we'll have this one recorded.
So we did get somebody ask, when was the corpse flower? When did it first come into notice by Western science?
1880?
1878.
There you go, 1878.
I was close.
When it was named as a species.
Oh yeah, Devin just shared it in the chat. Italian botanist Odoardo Beccari, 1878.
Yeah, and it's always, you always want to be mindful that the people who live in Sumatra knew this plant for, thousands of years prior to it being named a species in Western science. So this is a plant that, would have caught the attention of anybody who ever saw it, and they would be very familiar with it.
How many have we had bloom at the Botanic Garden?
Gosh, that is a good question. 10, yeah, I think, 10.
Close to 10.
We had 3 in 1 year, which was, kind of crazy, but really awesome.
Mm-hmm. Someone said they read the second plant is a clone of the one that bloomed in 2017. Can you talk about the cloning process or what that means exactly when we say it's a clone?
Well, just for reference, you actually brought up a picture of its parent, the one with the almost black-colored spathe. That is the parent, but we call it a clone because it's genetically identical. But actually, the way we propagated the plant was it was a vegetative offshoot of the corm. So it would have the corm would have looked similar to that, and you would have seen a second corm developing off to the side. And what we did about 4 years ago was split those two corms apart to become individual plants and grew them separately. So because it grew that way, it's genetically identical to the parent, which was the inflorescence that you saw in the picture there.
So would you expect it to look similar to that picture right here, this one, when it blooms?
We would.
Yeah, we would expect it to look extremely similar to that since they are genetic clones.
So far, so good. The spadix is a similar color, and that should develop as it gets bigger. But so far, so good.
Yeah, and the spade is starting to get little tiny purpley dots on it, so it's going to change the too from its green. The outside might stay quite green like that, but it's the inside that we expect to be really darkening up.
And Steve, I think you said this earlier, but just to reiterate, when do you anticipate the other one blooming or coming into peak bloom?
In about a week to a week and a half. When we see the spadix emerge out of the spathe, and or in this case, that bract you see here, it's withering away, was covering up the spadix. But as soon as we see that spadix for the first time, it's about a 2-week indicator that the plant will bloom. And that's give or take a couple days, but it's usually pretty reliable as a predictor.
Awesome. People are asking where the parent plant came from for the clone.
So these are siblings of each other. These were both, the parent material of this plant and this individual here were both donated as seeds from a gentleman in Hawaii. And we actually know the parentage of the seed. So, but they were both siblings grown from seed here at the garden in 2012.
Yeah, people may be familiar with the sort of what zoos do for parentage for animals. It's really important to obviously to breed animals that are pretty genetically isolated from each other, so that you don't get inbreeding suppression and that kind of thing. Making sort of mutant animals basically by doing that. And this is a case where we're really trying to kind of replicate that in the plant world, with really understanding the parentage of these plants that are in cultivation, and really just doing selective and very intentional breeding of these plants to maintain that genetic diversity in this sort of cultivated population. Yeah, Steve mentioned that bract. It's really spectacular. I don't know if you can see it in here, but yeah, I think you guys can see it. It's really amazing. These are modified leaves that really their function is to protect the developing flower bud, the developing inflorescence bud. And they did a great job in these two for sure. And once those, really fall away fully, we know that the bloom is pretty imminent.
Can you talk a little bit more, Susan, about the botany of these plants a little bit? Just about maybe the life cycle?
Yeah, absolutely. Yeah. So Steve mentioned the quorum already. So basically these plants have kind of 3 stages. They have a dormant stage, which looks very much like this, although usually it's under more podium. And this is the underground structure that is a storage unit. It's all stem tissue. And the plant will hang out in that stage for anywhere between a few months and really longer, up to a year maybe in some cases.
Yeah, and I've attributed that to conditions. And anecdotally, it seems like if you were to repot one in the summer, they tend to come back right away, whereas in the winter, when the conditions are less ideal, they'll kind of stay dormant for a while.
And then from there, it goes into bud, and that bud is either going to eventually produce a leaf, or it's going to produce an inflorescence. So here we have a picture of a leaf, and this looks like a tree. It's got a really large leaf stalk, which is called a petiole, but it looks like a trunk. And that trunk, if you will, the petiole of that big leaf there, has a lot of airspace in it. It sounds like a watermelon if you knock on a watermelon, and it sounds like that, which is really different than if you're knocking on a true tree trunk, obviously. So it's either going to produce that leaf. How long do our leaves usually last, Steve?
I've seen them go over a year, to be honest, but anywhere from 6 months to 12 months. I'm sure they could go even longer, but that's a good range, 6 to 12 months.
And then they'll start to senesce, and then Steve will cut those down, and they'll go back into a dormant stage. If that bud was actually for an inflorescence, then you can see what we get from that. And the inflorescences are going to take a few weeks to develop once we kind of get to the stage where we can tell it's an inflorescence. And then if we successfully pollinate it, that fruit takes, it can go pretty fast, and it can take up to a couple of months to really get nice fruits there. I think those took maybe 3 months to fully mature.
At least, yeah.
Might have been longer than that.
Yeah.
And then we'll usually plant those out, and then we often will share any seedlings that we get with other botanic gardens and with universities to add to their collections.
Susan and Steve, we got a question saying the seeds come from Hawaii, but the plants are not native to Hawaii, right? So where do the plants come from? Where are they native to?
The plants are native to Sumatra, which is one of the islands in Indonesia, and they only grow in Sumatra, actually, which contributes to their endangered state. Their range is very limited to a single island. So Sumatra and Indonesia, which is in Southeast Asia.
And I highly recommend watching the video that Steve did. It's on our website about the genus Amorphophallus. It's a great video that really shows some of the morphological diversity within that genus, which is obviously more widespread than just in Sumatra as a whole genus, but they're really beautiful examples that are a little easier to grow at home and much smaller than these giant ones.
Yeah, the Amorphophallus titanum are not great houseplants, correct? If you want to get them to bloom.
They are when
Or
they're
even
very
delete.
young, but you'll likely not have the ability to have one bloom at your house.
Do the relatives smell less than the titanums, or do they all kind of smell pretty bad?
There are relatives actually within our collection that I think smell far worse than the titanum. Eucyanus is a species that it's a very small plant, but the inflorescence maybe 6 to 8 inches tall. That I can't stand the odor of that one. That smells very much like the worst rotting fish you've ever smelled before. So, and capable of stinking up the entire greenhouse with a little tiny inflorescence. So.
And even the one that's kind of probably most commonly in cultivation and home gardens, konjac, that one is quite smelly as well.
Awesome. Well, I want to thank you guys. We're just about at time here. I guess if there's anyone else who has a question, they can throw it into the chat real fast. We might be able to take one more, but it's getting kind of late, and I want to make sure you guys have time to maybe grab some coffee before you do what you're going to do with the course flowers later today, get some pollination going or recording temperatures and whatnot. So thank you to Susan and to Steve for your time. Really appreciate it. And to Devin for helping run the Q&A in the chat as well. And we'll probably see everybody again real soon for more corpse flower fun. So thanks so much.